and now I don't want to talk too much and start with our first visit to the city of Leipzig in Eastern Germany, where the Fraunhofer Institute for Cell Therapy and Immunology is based. Dr. Dirk Kuhlmeier and Dr. Dave Smith will be explaining us which are the main research topics of the institute. But first, Keshia Aaliyah Aerchlimann will show us a little bit around and give us an insight to the offices and laboratories. So, curtain up. So now we are supposed to see the video. Yes.
Welcome to the Fraunhofer Institute for Cell Therapy and Immunology. The institute is located here in Leipzig and also in other locations in Germany, and we have about 650 employees. We also have large GMP facilities in our institute. Our core competence is the development of living cancer medicine in the field of oncology. Besides that, we also have unique capabilities in the field of medical diagnostics, ranging from biomarker research, bioinformatics and assay development to the development of diagnostic devices. Let me take you through the institute and show you what we're doing here.
Ja, alles super. Ich wollte jetzt eigentlich gleich nachher ins Labor. Dankeschön. Aber das ist später auch noch dazu, ja? Tschüss.
Welcome to a BSL-2 lab. I took you to this lab because the entrance to a BSL-3 lab, where we're working with viruses, is by far more complicated. In this lab, we are developing novel techniques for infectious disease diagnostics. Let me show you two different competences that we have here. Imagine you could detect an infection within minutes without the use of complex lab-based PCR tests. That is the vision we're working on using non-invasive techniques to analyze the breath of a person. With devices like this ion mobility spectrometer, we are able to distinguish bacteria from each other and to characterize antibiotic resistances. At this stage, we are trying to characterize COVID-19 infections directly in a patient's breath. However, this is a very big challenge because imagine the different substances you might have in your mouth just from eating, drinking, or even toothbrushing. The sample intake takes only 4 seconds, and the artificial intelligence-based analysis of the measurements is performed within 4 minutes. At this stage, the instrument is quite big, but we are not only working on the application of non-invasive diagnostics, but also on its miniaturization. Not everything can be covered by non-invasive diagnostics. That's why we are working on highly integrated lab-on-chip devices. Together with our partners, we're focusing on applications in areas of infectious diseases, neurodegenerative diseases, cancer, and liquid biopsy.
Sorry, Annika, you have to switch on your microphone, please.
Thank you for following our tour through the Institute. Our Fraunhofer Institute is always close to the industry. That's why we're looking for industrial and clinical partners presenting us with various challenges. If you are an academic partner, please contact us because our mission is to support the translation from basic research into products. We have great infrastructure to work with highly infectious pathogens, we have good manufacturing practice in place, and we understand the needs of tomorrow's industry. We are keen to work together to solve problems and to create innovative ideas for the future.
Thank you very much, Keshia, and thanks a lot, Caroline, for the introduction. Yeah, we have problems with Keshia because all the big movie companies are now after her, after that nice presentation. We would like to take you, my colleague Dave, who will join me as well tonight, we will take you from the scientific side in more detail into our institute. And let me see, let us start with a little bit of geography at the beginning. Does it actually work? Oh, it does. Can you see? Okay, the changes of the slides, that's important. Our location, as Carolina already mentioned, is in Leipzig, beautiful city. It's about 70 minutes by train south of Berlin. So it's a very center, like nearly in the center of Germany. And what I would like to do is I would like to take you to our Diagnostic Competence Center, because as you have heard, my name is Dirk Kuhlmeier and I'm heading the diagnostic department. So it'll be a focus more on the business unit of diagnostics at the IZI. As you see here, we are working and dealing with approximately now 80 to 100 people on these different areas like device development, diagnostics, and data analysis. And that ranges from biomarker research, for example, based on proteins, different proteins or RNAs, to bioinformatics, assay development and implementation, integration of assays in diagnostic devices, microfluidic structures, as you have seen in the video of Keshia, and non-invasive diagnostics. So take just one glimpse on an example here of our value chain, what we can actually offer. These two guys coming from industry, for example, they are eager to have a diagnostic device for the early detection of Alzheimer's disease. This is just one example of our developments here. So we can offer biomarker research, implement proteins, for example, which are already known as early markers for the Alzheimer's disease, bioinformatics, and the implementation of specific assays on point-of-care cartridges or microfluidic chips and build the device around that. So this is what we can do here at the IZI. And just to give you a quick example for the molecular diagnostics of infectious diseases, let's move to a really bad one. That's bacterial infection, which could lead to bloodstream infection, which could lead to sepsis, for example. Nearly 500,000 people in Germany are affected each year by nosocomial pathogens like multi-resistant pathogens, with like 15,000 people dying. And one reason is that the detection of these bacterial infections takes quite a long time. And here you see a kid with the mother seeing a doctor, and what they normally do at this stage is, up to now, they take blood in case of a bloodstream infection and they analyze it in standard lab environments, and that takes quite a long time. It can take up to days before you get a result. And for some, stick to the example of bloodstream infection, it's by far too long. So at the point of care, as you probably know, you take the sample in close proximity of the patient, you add it to a microfluidic cartridge, and this integrated cartridge can analyze the sample within minutes, and the doctors get information about that very, very fast, and they can decide whether another treatment or medication is needed. And these point of care tests are not only useful for COVID-19 detection of elderly people or in sports, but also with home care. So they are needed in many areas of our daily life. And sticking to the bacterial infection, I would like to show you just a quick example of a real-world sample. We have a bloodstream infection, which if you look at 1 milliliter of blood, we have something like probably 30 bacteria inside, but a large surplus of leukocytes having human DNA. And this, as you know, human DNA interferes significantly with a PCR. So if you have just pure, and here you see a qPCR signal that can be nicely done for bacteria. But if you have a real sample with human DNA, it looks like that. That's just 10% of the human DNA, which is normally in a milliliter of blood. So we have to find solutions in sample pre-treatment to get rid of the human DNA. And I would like to show you one old project, and there comes that microfluidic chip into play, what my colleague holds in her hand. We modify magnetic particles with specific binders, which can bind to bacteria in the blood sample. And these nanometer-sized magnetic particles, they will be transferred in the blood. And here you can see them. They are capturing specifically the bacteria. By using a magnet, you can transfer them to a microfluidic cartridge. You move them around, you lyse the bacteria, you do a PCR, and then you hybridize it specifically to specific capture probes, and you get a result without the influence of human DNA. That's made for a more clinical approach, but let's go further to home care molecular diagnostics. And one target is chlamydia infections and sexually transmitted, for example. And look at the numbers. More than 140 million people every year are infected by chlamydia. And you can see here the risk of an infection. But if you think of molecular diagnostics at home, how would I do PCR at home? You probably think of your heating. You need heating source, cycling of samples. Shall I do it in a microwave oven, or shall I use my gas or my central heating? That's not really doable. So what we are doing within the device is we have a chemical heating, a simple non-toxic chemical heating that you can create a temperature which is enough to drive isothermal amplification of the chlamydia nucleic acids. So this simple solution could lead to a device like that, presented by will be sold by the company here, Self-Diagnostics, called in Leipzig. You take this orange fin out and just take a droplet of urine, you sample yourself, and you can measure can use isothermal nucleic acid amplification to amplify just small concentrations of your pathogens in the urine. And you will get a signal visible by naked eye if you're positive or negative. That's just one example of what we are doing. Just go briefly to the non-invasive detection of diseases. I think I don't need to talk much about that because the BMBF decided like 4 or 5 years ago to give us some money for the development of a solution addressing the measurement of infectious diseases at airports in case of a pandemic situation. Now we have COVID-19, and it's a good example. We are not there yet, but what we are doing and what my colleague Keshia has shown already is the use of an ion mobility spectrometry. It's in the clinic at the moment. We test COVID-19 people within one breath for 4 seconds and an analysis for about 4 minutes. We can at the end of the day, hopefully distinguish COVID-19 from other diseases like influenza, for example. So if you're still interested, we can talk later if Dave is quick, yes, and I guess he's quicker than me. And for more information, just visit our booth later on. You're very welcome, or get in touch. That's no problem. Just one more thing I would like to add. That's a big funding we obtained here in Leipzig. It's called Diana, funded by the ministry, our federal ministry. The vision here is, in that area, we connect people with lots of money together to, speaking about our vision, we want to be the leading innovation and competence region for point of care in Germany and Europe. We have partners from all different areas which are needed to create a diagnostic device on the point of care. Partners, even international partners, are very welcome. So get in touch that we jointly offer solutions for future questions of medical diagnostics. So thanks to my co-workers here in our group, and this group is part of the diagnostic department, as well as my colleague Dave Smith, who is heading the DNA nano devices group. And Dave, please take over.
Okay, thank you. Carolina, about how long do I have? I'll tune my speaking speed to the amount of time we have left.
8 minutes.
Okay. Well, no, that's merciful for everybody in the audience. Okay. I hope, is everybody seeing my screen now? Is that visible? Actually, now it is full screen. Okay, cool. I'll just go ahead and jump right into it since we don't have a ton of time. So as Dirk mentioned, I'm working in his department. Actually, I've been in this department since 2013, running the DNA nano devices group. And I'll explain a little bit in the next slide what our basic technology is, but kind of the idea that I'm presenting is the subset of projects or kind of themes that we have where we're essentially using DNA or nucleotide materials to develop tools that then we start applying to different sorts of diagnostic and analytical applications with a little bit of fun therapy-based projects in there. So I could go on for actually hours, like literally I give university lectures on this, but very shortly, the basic concept behind our group is that we use DNA molecules not for their genetic code, but instead we're using it as a programmable construction material. You can see this picture on the upper left where you see this kind of nice four-armed looking structure. Let's say this is kind of the basis of the entire field of DNA nanotechnology, is this idea that you can take multiple DNA strands and make structures that are no longer like linear double-stranded DNA, but instead you can design your strands and have this concept of partial complementarity so that you can make branched structures that have 3 arms, 4 arms. You can also make hugely complex structures out of thousands of strands. I think maybe perhaps some of you have heard of techniques like DNA origami and DNA bricks. We actually work with that quite a bit, but I'm not going to be presenting this here. For us, what's very important about these DNA techniques is having the ability to attach bioactive ligands to them. You can see kind of the idea on the upper right, more or less due to the well-known structure of DNA and the fact that we can also order oligonucleotides very cheaply and with any sequence we want. What it gives us is a completely precise, like really molecularly precise structural template or scaffold that we can basically put bioactive ligands on, really with any sort of arrangement we want. You can see here on the upper right that you have, for example, 2 ligands on this double-stranded segment of DNA that would be 15 base pairs apart. That would be about 5 nanometers. But if we now want to move them closer, we just conjugate them to different parts on the DNA strands. And so we can really control, even down to, with really complex chemistry, down to sub-nanometer resolution, how we arrange different sorts of ligands. And so what this allows us to do is take advantage of these concepts such as templated multivalence, basically cooperativity between ligands that are joined on a single scaffold, and use these to enhance the activity of bioactive ligands like peptides and whatnot. I'll go into the next slide. And so this is something we've applied both to therapeutic areas, specifically with antivirals. You see this on the lower right, but probably what I'm going to focus on more here, the diagnostic and analytical applications. So a very important thing is what sort of ligands we use. We're actually quite flexible. Really, the only thing limiting us is kind of chemistry and then simple physics that I won't go into. But our preference, just from the topics we work in, are small bioactive ligands. We aren't a ligand development group, we're a ligand enhancement group. So what this means is that we already like to know the target and interaction. So this means we have partners that come to us with their favorite ligands. And then you can do some sort of chemical optimization depending on the type of ligand. So we do a lot of work with peptides and proteins. Our favorite things to work with actually are short peptide fragments that you can make in a synthesizer that are like, 5, 10, maybe 20, 25 amino acids long, which we can use to target surface receptors on cells, viruses, bacteria, or even other proteins like antibodies, things like this. For the last few years, we've also been working with glycan or sugar molecules, carbohydrates, namely synthetically produced oligosaccharides. They're actually hugely complex to make, as I found out when we started working on this, but they're really nice little ligands for interacting with things like bacteria and viruses or sugar binding proteins known as lectins. We also work with what I like to term as big stuff, which is basically just means we put DNA strands on things like antibodies or enzymes, or nanoparticles or inorganic nanoclusters. So we're really flexible there with what kind of ligands we can put on our DNA constructs. So then the next thing is, what do we actually do with them? Well, our main areas, let's say more in the diagnostic side, or let's say the non-therapeutic side, are looking at either diagnostic systems. Dirk kind of introduced some areas before, for example, like these lateral flow things. This is something we're actively developing with his group, but also we've applied this to different sorts of immunodiagnostic systems where we're detecting antibodies that might be indicative of some sort of a disease or immunity. Where we've come actually, let's say the farthest when it comes to translation for commercial applications are within putting these in analytical systems. What this means, and kind of one of the papers you can actually download in our booth and the networking session, we have integrated this into drug discovery platforms that are based on binding between molecules. You can think of this like surface plasmon resonance or some of these other sorts of assays, but also we apply these as labels for flow cytometry, or we work with one company that does what's called chip cytometry, and really anything that's related to binding a specific target. We also work with some therapy-related applications, but I won't go into those. However, I can, again, if you come by our booth later, I can point you towards a paper where we actually do this in a nice way. One of our big focuses is on interacting with virus surface proteins. The concept here, I don't want to go too into it because, again, it's a very complex topic I could talk a long time about. But the idea is that virus surface proteins, for example, the hemagglutinin receptor or the well-known spike protein on SARS-CoV-2, typically have a very well-defined multimeric structure. And so what this allows us to do is design our DNA constructs in a way so that it presents the virus-binding ligands, whether it's a carbohydrate, sugar molecule like a sialic acid, or a peptide, which has been developed, and we've actually done this in one instance, to block the fusion of the virus with the host cell. We can arrange these ligands on DNA structures that greatly enhances their activity. And again, this is both used for viral prophylactics. That was kind of our entry into this game. However, where we found a lot more success in terms of what companies are interested in is using this to develop drug discovery platforms that are based on kind of binding. So people that want to develop their own viral inhibitors and want to have a way to test them kind of on the natural virus structure, this enables the possibility to do that. Another thing which we do, because one of the ideas that we want to do is let's say, effectively democratize this complex method of DNA nanotechnology. Basically, a lot of the techniques which are always used are things like TIRF microscopy or SPR, things which require big, expensive, specialized equipment. Again, for those of you that are familiar with DNA origami studies, there's a lot of very expensive equipment which people have to use to really maximize their potential. So one of our goals that we've done over the last few years or achieved is we really wanted to take this complex DNA nanostructuring concept and then bring it to kind of a universal assay, which anybody can do. And we've done this most effectively using kind of a standard ELISA. I think in any molecular biology lab, or really most biolabs, you're going to have an ELISA reader, a plate reader. So everybody has the ability to do ELISAs. And so what we've done is we've integrated in many, many different cases these DNA nanostructures, either things like DNA origamis, where we can put hundreds of ligands, or let's say up to 100 to 200 ligands, or small DNA constructs like you see on the right that we would use to bind the surface of viruses. So we've put this into ELISAs. We call it the nucleotide templated ELISA, or Nutella for short, just to make everybody think a little bit of I don't know, one of the favorite breakfasts here in Germany, very tasty stuff. And so what this allows us to do is really just do a standard ELISA, which again, anybody can do, but now we can look at these effects, for example, like valency, nanometer scale or sub-nanometer scale arrangement on the effects on binding. We've done a lot of really nice studies here using a lot of different ligands like peptides, glycans, small molecules, and we've applied this with applications. We've done this in applications involving viruses, so whole viruses like influenza or SARS-CoV-2, viral proteins, lectins, antibodies, recombinant cell receptors, and even in one instance, we worked with whole cells. So it's really, really cool technique, we think. The other thing which we're working on, again, is working on making synthetic DNA peptide constructs that we use for cell surface labeling. This is going more in the direction of cytometry or histology. We're also very heavy on flow cytometry. And so this is actually something we're very active in, is really designing optimally structured DNA constructs that kind of, let's say, replace antibodies in flow cytometry. There's a lot of different functionality that we can get with them, but I won't go too much into here. And then also one thing that we can do, which is a lot of, again, this came out of another project, but we discovered this is kind of something that works quite nicely, is we can also make these synthetic DNA constructs which specifically target structures in cells. This is very early work, so it's something that we're still developing, but for example, we published some constructs in 2018, which are specifically targeting the actin filaments within cells. Actually, the paper itself was about modulating the mechanical properties of actin networks that are taken out of cells. So actually, my own background is in biological physics, so we like to do a lot of biophysics in the group. But one day we decided, hey, why don't we put these in cells? And what we found is actually these bivalent constructs bind very nicely to actin filaments. And so they label actin filaments and stress fibers and also the actin cortex quite well. So we're also beginning to develop labels that we can use really for classic cell labeling for assisting in microscopy. And with that, I think hopefully I haven't gone too much over the time. And hopefully, in contrast, I didn't speak too horribly fast. But with that, I'm going to go ahead and close out. And what I'll just say is that if anybody has questions for me or for Dirk about our institute or also my group's work, what I just presented, I know I'll be in the virtual booth in about, an hour and a half or so when the networking session opens. And with that, I will close my presentation and give the control of the screen back to all of you.
Thank you so much. This was very, very quick, but I think the experts, understood everything. Great. Thank you so much to both of you. I want to open the audience. I want to give the audience the word. So if anybody has a question, please feel free to ask Dirk or David. Just switch on your microphone. Yes, I see there's Jürgen Drescher wants to ask something.
Thank you very much, both of you, for this really very interesting talk. I want, in our networking event, maybe we can join tonight because we at the DLR, German Aerospace Center, we are not doing the same methods like you, but we have the same applications. You pointed out you have this so-called airport pandemia, the BMBF-funded project. You have this non-invasive detection of diseases. So please let us talk afterwards so that we could bridge and create a win-win situation. Thank you very much.
Sure.
That would be nice.
That would be great.
Great. Thank you. Is there another question? Or is there a question, actually?
Otherwise, I will ask Dave a question.
Sure.
Feel free.
No, just kidding. Just to put some pressure on the others,
I have, of course, prepared questions, but I'm sure we have time afterwards. So for everybody who has joined later, at 7 p.m. of German time, we will be finishing, and then you have the opportunity of getting in touch at the booth. So feel free to talk there. So I would just say thank you so much for your video, also to Keshia, who's not here, who made the video, and I will now go over to the next presentation